besties! In this video, we're going to be discussing the top 15 common questions that you're going to see when it comes to the ATI TEAS Science, Life, and Physical Science portion of the exam. Starting with our first question, which of the following is responsible for carrying amino acids to ribosomes during protein synthesis? And the correct answer is going to be D, tRNA. So tRNA is actually found down here towards the end of our scale. And what does it do? What its main responsibility is it needs to carry amino acids to the ribosome during protein synthesis. What it does, and let me zoom in because I think this will help you visualize it, is what it does is it reads this bottom layer right here, this bottom layer, and it's going to have the anticodon to the codon in our bottom layer, and that's going to be attached specifically to amino acids. And as it goes down reading all of the information on that piece of RNA that was synthesized, it's going to keep attaching amino acids, so on and so forth, until we have a completed protein chain. But let's look at our other options. So we have A, mRNA. So mRNA is actually found right here at the beginning. And what it does is it's really the messenger, right? It's in the name, messenger RNA. It's going to carry that genetic instruction from the DNA to the ribosomes. And the ribosomes are ultimately going to be doing the coding sequence when it comes to the protein synthesis. Like we just discussed, B is also incorrect because it's ribosomal RNA, rRNA. It's going to take the information that it got from the messenger RNA, and it's going to start to look at it, make sure that there's no errors in the code, make sure that it is a good piece of sequence that can be translated into amino acid protein chains later. And then lastly, we have C, which is just DNA. DNA contains genetic information for protein synthesis, but it's not directly involved specifically in the carrying of amino acids to the ribosomes, making it also an incorrect answer. What is the primary role of the sodium-potassium pump in animal cells? Does it generate ATP? Does it maintain the cell's osmotic balance? Does it allow molecules to enter the cell? Or does it transport carbon dioxide out of the cell? And you guessed it, the correct answer is B. It helps maintain the cell's osmotic balance. So like we've talked about before in many of our ATI TEAS lives, if you have been watching them, I highly recommend that you join us every Monday, Tuesday, and Thursday at 7 p.m. Eastern Standard Time when we talk about different concepts. But like we've said before, sodium and potassium have a very inverse relationship. They don't like each other. They're like exes, right? They're like your ex. You don't want to see them. And that's what's happening with sodium and potassium. If we've got too much sodium in the cell, potassium's going to get pumped out. If we've got too much potassium in the cell, sodium's going to get pumped out. And that's what helps maintain our osmotic balance and proper ion concentration in our body. Looking at other options, we have A, to generate ATP. We know that that's actually done in the mitochondria, and that's actually part of a process known as cellular respiration, not the sodium-potassium pump. Then we have C, it allows molecules to enter the cells. This would actually be done by some kind of channel, right? We have aquaporin channels to allow water into cells. We have protein channels to allow certain other kinds of ions to get inside the cells. We've got different kinds of channels in order to make this happen. This is not happening in the sodium-potassium pump. And then lastly, to transport carbon dioxide out of the cell. Carbon dioxide is a waste product, specifically of cellular respiration. And it actually diffuses out of the cells using passive diffusion. It does not need the sodium-potassium pump in order to make that happen. All right, buckle up, Buttercup, for our next question. What is the difference between a monosaccharide and a disaccharide? Is it that monosaccharides are composed of 2 simple sugars, while disaccharides are composed of more than 2 simple sugars? Is it monosaccharides are simple sugars that are made from amino acids, while disaccharides are made from fatty acids? Is it disaccharides are composed of 2 simple sugars, and monosaccharides are simple sugars that cannot be broken down into simple sugars? Or is it monosaccharides contain more carbon atoms than disaccharides? And the correct answer for this one is going to be C. Disaccharides are composed of two simple sugars, and monosaccharides are simpler sugars that cannot be broken down into even simpler sugars. So think about it. Monosaccharides are the simplest form of sugars. It's actually considered the monomer of carbohydrates. Mono meaning one, saccharide meaning sugar. It is the simplest form. You see them in things like glucose and fructose. In contrast, we have disaccharides like sucrose, which is actually a form of table sugar, and even lactose. And they get their name di, meaning two, meaning that we're going to have two sugar molecules combined together, making C the correct answer. Looking at other answer options, we have monosaccharides are composed of two simple sugars, while disaccharides are composed of more than two simple sugars. We know that that is incorrect because monosaccharides only have one, and disaccharides shouldn't have more than two. Then we have monosaccharides are simple sugars that are made up of amino acids, while disaccharides are made up of fatty acids. This is also incorrect. Monosaccharides are carbohydrates, they're sugars. They're not made from amino acids. And disaccharides are made up of two monosaccharides, also not fatty acids, making B incorrect as well. And then lastly, monosaccharides contain one carbon atom, then disaccharides. Again, incorrect. The number of carbon atoms in monosaccharides and disaccharides can vary greatly, but the key distinction lies in the structure. Simple sugars versus two sugar combinations, not how many carbon atoms they have. Which of the following statements best describes the process of meiosis? Is it the result in the formation of two genetically identical diploid cells? Is it responsible for reducing the chromosome number in gametes? Does it occur in somatic cells to produce identical daughter cells? Or does it involve the division of the nucleus followed by cytoplasmic division? And yes, the correct answer is B. It is responsible for reducing the chromosome number in gametes. Remember that word gametes is synonymous with meiosis because gametes is just a fancy way of saying our sex cells. So that's our sperm and our egg cells. And what they do is they take half the chromosome number from one parent, half the chromosome number from another parent, and they combine them together, ensuring that we have genetic diversity in the offspring. Looking at our other answer options, we have A. It results in the formation of two genetically identical diploid cells. This is a fantastic example of mitosis. Remember, in gametes, we're going to have genetically diverse cells, whereas with mitosis, we're going to have two genetically identical cells. Then for C, it occurs in somatic cells to produce identical daughter cells. So somatic cells is synonymous with the word mitosis. Somatic cells just means it's our body cells. So if we were to get a cut on our arm, what would happen is those somatic cells would go through the process of mitosis to create identical surrounding cells in order to hopefully not leave a scar on our skin. However, if we had meiosis, since they're genetically different, then that means we would have a really weird, funky looking wound that would occur because it doesn't create the same kinds of cells. Somatic cells create the same cells, they're body cells, and gametes create genetically different cells, they are meiosis. And then lastly, it involves the division of the nucleus followed by cytoplasmic division. Again, not really what's happening specifically in meiosis. We do have the division of cells, but it's not the main reason. It's not the primary thing that's actually occurring here. And that is the reason that D is also incorrect. So what is the name of a genetic disorder caused by the presence of an extra chromosome 21? Is it Down syndrome, cystic fibrosis, Huntington's disease, or sickle cell anemia? And you've got it. The correct answer is A. It is Down syndrome. Down syndrome is caused by the presence of an extra chromosome known as chromosome 21. And this results in a total of 47 chromosomes instead of the usual 46. With this extra genetic material, it's going to affect physical and intellectual development of the offspring. So with B, we have cystic fibrosis, and this is actually a genetic disorder caused by mutation on one specific gene known as CFTR gene. And that gene stands for Cystic Fibrosis Transmembrane Conductance Regulator. It's a specific protein that helps maintain the balance of water and salt on many surfaces found inside of our body. With C, we have Huntington's disease, and this disorder is caused by a mutation of our ATI gene. And this leads to a progressive neurodegeneration not caused by an extra chromosome. And then lastly, we have sickle cell anemia. So this is actually a genetic disorder in the blood. It's a mutation of our hemoglobin gene. And this will lead to very abnormal shaped red blood cells. They almost look kind of like a half moon. And it's very, very uncomfortable for those individuals that have this disorder. So which of the following describes an individual with a genotype capital A, lowercase a, capital B, lowercase b, where the capital A and lowercase a represents alleles for one gene, and the capital B, lowercase b represents alleles for another gene? Is it that we're going to see homozygous for both traits, heterozygous for both traits, homozygous for one trait and heterozygous for another trait, or heterozygous for one trait and homozygous for the other? And you've got it, the correct answer is B. They're both going to be heterozygous. So remember, whenever we have an uppercase letter and a lowercase letter attached to each other, that means that we're going to see a heterozygous genotype, hetero meaning different. However, in contrast, if we maybe had two uppercase letters or two lowercase letters, then we're going to see homozygous genotypes, meaning homo means same. You could have a homozygous dominant, two uppercase letters, or homozygous recessive, two lowercase letters. So if you look at both of our genes, we have an uppercase, a lowercase, and another uppercase and a lowercase. That means that all of our traits are going to be heterozygous based on what we just discussed. In order for them to be considered homozygous like we see in option A, like we talked about, they would either have to be both dominant or both recessive. In this case, we have both heterozygous genes, making A incorrect. And C and D kind of follow that same thing, meaning that one trait would be homozygous and one trait would be heterozygous. They just kind of switched it, so it's kind of the same answer, just said a little bit differently. But if one trait was maybe like capital A, and then another trait was maybe capital B, lowercase b, then these would probably make sense, but that's not what the question is specifically asking. So in this case, they would both be incorrect. So which of the following microorganisms lacks their own metabolic pathway and can only reproduce inside of a host cell? Is it bacteria, fungi, virus, or protozoa? And you're absolutely correct. The correct answer is C. It is a virus. Remember, viruses lack their own metabolic pathways. They can only reproduce by infecting host cells. If you get a virus inside of your body, it doesn't do anything unless it comes into contact with your cells. That's the only way that it can reproduce. So we call this type of reproduction obligate intracellular parasites. Looking at our other answer options, we have A, bacteria. Bacteria is its own independent microorganism. They have their own metabolic pathways. They can reproduce on their own without the need of infecting your host cells. So that's why we need things like antibiotics to combat a lot of these bacterial infections, because they are able to do a lot of damage on their own. Fungi the same way, right? They are eukaryotic organisms with very complex metabolic pathways. With these guys, if we get a really bad fungal infection, we're looking at antifungals to help us beat what's happening inside of our body. And then lastly, protozoa, same thing. They're single-celled eukaryotic cells. They have their own metabolic pathways they can produce independently without the need of your host cells. Which of the following best represents Mendel's law of independent assortment? Is it alleles for a gene are inherited together because they're located on the same chromosome? Is it the inheritance of one gene does not affect the inheritance of another gene located on a different chromosome? Is it a dominant allele will always be inherited over a recessive allele? Or is it both alleles for a gene will be passed on to offspring? And the correct answer for this is going to be B. The inheritance of one gene does not affect the inheritance of another gene located on a different chromosome. Remember, we talk about Mendel's law of independent assortment. It states that genes from different traits are inherited independently of each other if the gene are of different chromosomes, which is the reason why B would be correct. Looking at other answer options, A, alleles for a gene are inherited together because they're located on the same chromosome. This would actually refer to a process called gene linkage, not independent assortment. Then we have C, the dominant allele will always be inherited over a recessive allele. This is actually an example of something called dominance, not independent assortment. And then lastly, both alleles for a gene will be passed on to the offspring. This is not a description anywhere when it comes to the law of independent assortment for Mendel's law of inheritance. So which of the following is used to determine a person's DNA sequence? Is it genes, ribosomes, enzymes, or chromosomes? And the correct answer is going to be A, genes. Remember, genes are segments of DNA, and it contains very specific code for specific proteins. That sequence of nucleotides that are found within the gene help us determine the amino acid sequence of proteins that it needs to encode. So absolutely, when we're trying to determine a person's DNA sequence, it's going to be the genes of that sequence, making A the correct answer. Looking at other options, we have ribosomes. So ribosomes are the structures that help us translate mRNA. It has nothing to do with determining DNA sequence itself, making it the incorrect answer. Then we have enzymes. Enzymes are great proteins when it comes to catalyzing chemical reactions, but again, doesn't have a whole lot to do with the DNA itself when we're looking at the sequence. And then lastly, we've got those good old chromosomes. So chromosomes are structures within cells that contain DNA. However, a chromosome is composed of many genes, not just one. So that is the reason that D is also the incorrect answer when it comes to determining a person's DNA sequence. So the cytoskeleton of a cell is composed of which of the following macromolecules? Is it carbohydrates, nucleic acids, lipids, or proteins? And the correct answer is going to be D, protein. So what's cool about the cytoskeleton is it's made up of protein filaments known as microtubules. You also have actin filaments and intermediate filaments, which all provide structural support, shape, and help facilitate cell movement. When it comes to carbohydrates, they're primarily used when it comes to energy storage and structural purposes like in cellulose found in plants. They're not the main component when it comes to the cytoskeleton. Nucleic acids, as you've guessed it, have a lot to do with our DNA and our RNA. They're not really looking at the structure of the cytoskeleton. And then lastly, those good old lipids, they help with things like forming our membranes, and they also help with storing energy. Not a whole lot to do with structural elements. What is the phenotypic ratio of offspring in a monohybrid cross between two heterozygous individuals for a single gene with complete dominance? Is it 3-to-1, 1-to-2-to-1, 1-to-1, or 2-to-2? And you guessed it, the correct answer is going to be A, 3-to-1. So what is this specifically asking? We know that we have two heterozygous individuals, which is why we have our two heterozygous parents on each side of our Punnett square. And we're looking for a single trait with complete dominance, meaning that the trait has to have a capital A in the trait in order for it to be dominant. So when we talk about phenotypes, we're going to talk about what is actually visualized in the offspring when they are created. So there's 4 potential things that we could see in the offspring. Number 1, we could see somebody that's just dominant, right? They just have 2 capital A's. We have 2 situations where we could have one person with a two possibilities where we're going to have a capital A and a lowercase a, and then one possibility that we're only going to see recessive genes. So if we were looking at the phenotype of this individual with complete dominance, 3 of our 4 squares tells us that we are going to be exhibiting dominant traits. If we've got a capital A, doesn't matter if we have the lowercase a, we're only going to see the capital A because it's dominant. So that would make A the correct answer, meaning that there is a 3-to-1 chance of our phenotypic types of offspring. Looking at our other answer options, we have 1 to 2 to 1. So this is actually our genotype ratio, where we have one genotype that's completely dominant. We have 2 genotypes that carry both a dominant and recessive allele, and 1 genotype that's completely recessive. So then we have a 1-to-1, meaning like we would have a 50-50 kind of situation of seeing either one. So this isn't typical of a monohybrid cross. This would be more related to if we saw something with codominance or even like incomplete dominance type of situations. And then lastly, 2 to 2, that does not apply when it comes to standard Mendelian monohybrid cross. So that would make D also an incorrect answer. So which of the following represents the complementary strand of a DNA sequence? So this is our original strand. We have 3 TCG, ATC, GCA5. So what would be the complementary sequence out of the 4 options that we have available to us? So the correct answer for this one is going to be C. Let's figure out how we would figure this out. So when we are looking at pairing our nucleotide bases, we have a couple different ways you can remember it. When it comes to DNA, we have apple in the tree. So A combines with T, adenine to thymine. Now if we're looking at an RNA sequence, then we know that adenine combines with uracil, meaning apples under the tree. So that's the way I like to remember the differences between DNA and RNA when it comes to our A, T, and our U. Now with our cytosine and our guanine, the way I like to remember it is car in the garage. So cytosine is always going to pair with guanine. In this case, because we have T's, we're looking at DNA sequence, not RNA sequence, and this is how we figure it out. So first I'm going to write everything out that we have up here, and now we're going to pair it with our complementary strand. So we're just going to do the opposite, or we're going to do the base pairing depending on what our letter is. So our 3 is actually going to be our 5. Our T is going to attach to our A. Our C is going to attach to G. Our G is going to attach to C. Our A is going to attach to T. Our T is going to attach to A. Our C will be G. G will be C. C will be G, and then our A will be T, and then of course this will be 3. So if we were looking at all the options we have available to us, C would be the correct answer, making it our complementary pair. Which of the following genotypes represents an individual who is homozygous for a recessive trait? Is it going to be capital A, capital A, capital A, lowercase a, lowercase a, lowercase a, or capital A, lowercase a, capital B, lowercase b? And as we discussed previously, the correct answer is going to be C. In order for us to have a homozygous recessive trait, that means 1, the letters need to be the same, right? And 2, they're going to have to both be lowercase. So C meets that qualification in order for us to find the correct answer. Looking at the other ones, we have a homozygous dominant with A, capital A, capital A. With B, we have a heterozygous genotype. And with D, we have 2 heterozygous genotypes. So a patient goes to the doctor with a sore throat and a cold. They ask for antibiotics, but the doctor refuses to prescribe it to them. What kind of microorganism is causing the illness? Is it bacteria, fungi, protozoa, or virus? And the correct answer is going to be D, virus. Like we discussed in a previous answer, a lot of times when you see sore throats and colds, they're usually viral in nature, and antibiotics are not going to respond to them. Antibiotics are effective against bacterial infections, not viral infections. Looking at our other options, if we had a bacterial infection, they were most likely looking at an antibacterial. If we had a fungal infection, then they would most likely get an antifungal. And then when it comes to protozoan infections, there's very specific medications that you would have to get in order to treat those kinds of infections. Antibiotics would not be one of them. And to be honest, protozoas don't typically cause sore throats and cold symptoms. Genetic information that describes characteristics of an organism is found in which of the following? Is it the cytoplasm, the nuclei, the mitochondria, or ribosomes? And you are absolutely correct. The correct answer is B. It is going to be the nuclei. That nucleus contains all of our cells' genetic material, also known as DNA, and it is responsible for storing and transmitting genetic information. Looking at our options, we have the cytoplasm. Cytoplasm is involved in cellular processes, but it's not involved with the specific storage of genetic information. The mitochondria, we all know that is the powerhouse of the cell. It helps us create ATP, not DNA. And then lastly, ribosomes. Ribosomes are really important when it comes to protein synthesis, but it doesn't actually store any of the DNA material, making B our only correct answer. That's all, besties. These were the 15 most common questions when it comes to life and physical science. If you want additional practice questions, I highly recommend that you check out this playlist. It is full of just really good information when it comes to the TEAS. As always, I'm going to catch you in the next video. Bye!